in this part of the difference between the structure and function of the RNA, I will show you the pictorially the DNA, the differences. Here you can see the DNA has a deoxyribose sugar, which is a hexose sugar. Here you can see the phosphate in the yellow circle, and here in the second in RNA you can see a red circle with OH, that is, it is a ribose sugar. It has got an extra oxygen which is not present in the DNA. Now the nucleotides which are present in the DNA are thymine, adenine, cytosine and guanosine, and in the RNA uracil, adenine, cytosine and guanine. The thymine in the DNA has been replaced by the uracil. Here in this you can see that the DNA is a double helical structure, whereas the RNA has a single strand. You can see the 3' end of the pentose sugar on one side, and on the opposite the 5' end. And in the double helical structure of the DNA, you can see on the opposite side is the 3' end, and the lower side you have got the 5' end. Whereas where the RNA is concerned, you can see it starts from the 3' end to the 5' end. In the next slide, again, you can see the single stranded structure of the RNA with cytosine, guanine, which has got 2 rings, adenine again has 2 rings, and uracil has got a single ring. And here you can see the base pairs, the double stranded DNA strand, and cytosine, single cycle, guanosine double cyclic, adenosine double cyclic, thymine single cyclic nucleolus bases. The structure, there are 3 types of messenger RNA, 3 types of RNA. The first is the messenger RNA. Messenger RNA carries copies of instructions for the assembly of amino acids into proteins from DNA to the rest of the cell, serves as a messenger. Here you can see a DNA and its corresponding mRNA coding strand of the DNA, and the mRNA corresponding coding is being done. Now the second RNA is the ribosomal RNA or rRNA. This makes up the major part of the ribosomes, where in proteins are synthesized or made. Here you can see an electromicrograph picture showing the ribosomal RNA. Then you have got the transfer RNA or the tRNA. The tRNA transfers amino acids to ribosomes during protein synthesis. Here you can see the structure of a tRNA very clearly. The structure, which is red in color, is the clover leaf structure, and here the tRNA has got an anticodon site wherein the codon of mRNA is attached, and you can see the amino acid site where the amino acid is attached. The proteins are made up of a chain of amino acids. Proteins are enzymes which catalyze and regulate chemical reactions. There are two steps to make a protein: transcription, that is DNA makes RNA; translation, that is RNA makes protein with the help of chain of amino acids, as seen very clearly from this diagram. Here, the second diagram, that is translation. In the first diagram, in the transcription, you can see that DNA manufactures the mRNA. Then mRNA translates the message of the DNA to the proteins. Here you can see the tRNA brings in the amino acids according to the codes. When transcription needs to take place, DNA must provide the code in order to create an mRNA strand. mRNA strand will be able to leave the nucleus, and now it has the code transcribed inside its base pairs. Thank you.